Welcome to the third basic lecture for the Deep Learning for Microscopy course. So in this lecture, we'll now discuss the combination of machine learning and image analysis after we've covered these two topics in the previous two lectures. So let's recap what we did in the last lecture. We segmented the nuclei with a thresholding-based approach, and you can see the result we have, where we have each nucleus with a different color here on the right-hand side. And you also see the problems that we have with this, right, where we had sort of tucture nuclei that were wrongly put in the same object as we see here and here and here. And so this was a problem with the thresholding approach that we've taken. And what we want to discuss today is how we can use machine learning to solve this instead. And what we discussed in the last lecture was quite an easy problem. So the nuclei were fairly simple to separate from the background just based on their intensity. But if we look at more complex segmentation problems, like segmenting mitochondria in EM over here, or segmenting cells in phase contrast microscopy, we can already see that even separating foreground from background, if we just want these objects or want to separate them, will be quite a bit harder than segmenting these nuclei in fluorescence microscopy. So this means we need more powerful methods for the segmentation task that don't suffer from these issues we see with the thresholding. And this is where now machine learning comes in, right? So instead of defining these thresholds or other rules, we want to learn how to segment objects in the image from examples. And you might say, well, maybe we can work on the thresholding a bit and make it cleverer and take into account other aspects of the image, right, to separate objects and to define what is foreground, what is background, right? Because what we've done so far is basically just look at the intensity of one pixel, right? We basically, for each pixel, ask the question, is this white, right, or what is its intensity? And then we apply a threshold based on this. And if the pixel is over the threshold, we say it's a nucleus. And otherwise, well, we could say it's not a nucleus. But we've seen then we run into this problem, right? That we have touching objects where we have sort of barely any space between the objects like here. So if we just apply the simple criterion, we cannot separate them from each other. So we might also look at the neighbors of the pixels to define what is in the nucleus and what is not in the nucleus, or which pixel is foreground. We might look at edges, right, and try to use this to define that something that's above the edge is not a nucleus. If we do this, we see that this also will not be enough. So we might look at texture and so on. And as you see, right, what we are back to is trying to come up with this intricate set of rules to solve a problem in our image. And we've already discussed this in the first lecture when we discussed the example for furniture classification, that finding good rules and generalizable rules for solving image problems is very tricky, right? So we don't want to go down this road, and we want to learn this decision instead. Similar for the very sort of simple toy problems that we've seen in the first lecture, we now want to apply machine learning methods to solve the image segmentation task that we are given here. So right, in sort of a high level, this would look like this, right? We start from our image, and then we compute features for this image, right? So, and these features are the inputs to our machine learning algorithm. And in particular, these are filters we compute for the image, right? So one filter could be something that highlights the edges, one filter that smoothes the image, one filter something that highlights different aspects of the texture. And we've seen in the last lecture, right, how these image filters are defined and computed. But the idea is here, right, that we combine these different filters that highlight different aspects of the image, and we take all of them as the input for our machine learning algorithm so that it can learn the decision of what is foreground and what is background, right? So what is nucleus and not a nucleus, which is shown in blue and yellow here, so that this algorithm can then learn this from training data. Right? And the training data would be annotations that we or the user provides in order to tell what is nucleus and what is not nucleus. So this is how this works on a very high level. Right? If we map this back to the machine learning scheme that we've already seen in the first lecture, right, we can sort of think about these features defining this sort of space that on one axis defines the intensity, for example, of our image or of parts or pixels in the image, and on the other axis defines the texture, right? And these are these features that we get from image filters. And then if we want to do machine learning here, right, we need annotations, as we already discussed. So basically we need some human to tell us, okay, this pixel here, the ones marked in blue, these are background, and the ones marked in yellow, these belong to the nucleus, so are foreground. And then we would label the corresponding points in our feature space, right? So basically we'd find, okay, the yellow labels here from the nucleus, they would be somewhere over here. The blue ones, which correspond to the background, somewhere over there. And then we apply a machine learning algorithm, as we discussed it in the first lecture, that finds a separation between these two classes. You know, in practice, this feature space would look quite a bit more complex, but you can again think about finding this line or decision surface that separates these classes. And then once we've trained this based on the annotations, we can then apply it to all the data, right? So we will know for each pixel if it's background or if it's foreground, based on what was learned by the machine learning algorithm that we use. So this is how we apply this concept of machine learning to the image segmentation problem in principle. So let's make this more concrete and give the example of a software that can do this for us, which is Elastic, which is a software tool for interactive machine learning, and in particular also for these segmentation tasks. So the way this is done is by Elastic, that in the first step, it computes these image filters that we use as input for the machine learning algorithms. It then uses user annotations for the pixels as training data, and then trains a random forest model to solve the segmentation task. Right, and if we go through these different steps, right, so we've seen the image filter concept in the second lecture, right, and so basically in Elastic, you can select all kinds of filters that are applied for the image. And these, right, sort of are these smoothing filters. These we already discussed. These are also edge filters and then texture filters, but basically these highlight sort of different aspects of the image. And you can see these smoothed or edge filtered or texture filtered examples down here. And we then have the user annotations, and the learning algorithm or machine learning algorithm that's used is a random forest, which is just an ensemble of decision trees, right? So remember, we discussed decision trees in the first lecture, and now a random forest is just a machine learning model that combines many of these trees and is thus more powerful than using a single tree. But the way this is trained is that as we've outlined already, right? We kind of stack together these different features or filters and use this as input to our machine learning model, right? So this is kind of the input or features. And then we have the annotations from the user, right? Which are basically these brush strokes that tell us for the pixels under the brush strokes if this is foreground or background. And then based on this, we train this model and can apply it to the rest of the data. Yeah, let's show how this looks in practice now. Just as a little video demonstration. So this is how this is in Elastic. We now have selected 3 classes: background, foreground, and boundary. And now, as you can see here in the background, I annotate the data with these classes, right? So now the background label in this example is yellow, so it's switched in comparison to before. The foreground label, so for the nucleus, is blue. And what I do right now is annotate the blue classes. And we get this automatic update. So you see now I select the boundary class. I draw the boundary here between these 2 touching nuclei, and we have this live update activated. So this means, we get the changes instantly of the segmentation algorithm. And what went through a bit fast was then the final segmentation result. So basically what the algorithm would predict for all the pixels based on our annotations. You can see this again here as a still picture, right? And so this is now, given the training data that we've provided, and again, training data were just these annotations for a couple of pixels, we now see the segmentation output for all pixels, where yellow here corresponds to predictions for background, blue are the predictions for the nucleus, and red are the predictions for the boundary in between the nuclei. And we now chosen here to use these three different classes, the background, the foreground, and the boundary, to better separate the objects. Because the boundary now allows us to also segment touching objects. You see this for this example here. So these are these two touching nuclei we've seen a couple of times. And we see that they are sort of foreground label. The blue pixels are not touching anymore because we have the boundary label in between. And now if we apply the connected component algorithm that we've seen in the second lecture too, just to this foreground label, so just connected component to kind of all these blue pixels, then we get this result. So we get these segmented nuclei, and we see now that the nuclei that were touching beforehand are now separated, because we made use of this boundary label here, also some examples here on top were touching, and we see now that they get correctly separated. So this means we now combined the machine learning that allows us to easily sort of train these three classes and train these boundary labels that separate its nuclei from each other where they should not touch, and then combine this with this classical approach or this image analysis approach of connected components that we know from the last lecture. And now we solved our problem of touching nuclei and have a correct segmentation. So we could now go back to this, measure the sizes, and we wouldn't get these errors in the size histograms due to the touching nuclei. Yeah, but we can also solve other problems here with machine learning. So we looked at the image here of the nuclei for quite a while already, and you probably noticed that there are two different kinds of nuclei in there, right? So we have here kind of the nuclei that are shown now with a yellow arrow, which have a bit dimmer intensity and are a bit bigger. And we have these nuclei here shown with a blue arrow that are a bit smaller and have a brighter intensity, right? And so the nuclei shown with a yellow arrow, these are just kind of the normal nuclei, right? So for a cell that's in a normal cell state and is still living. And the blue arrows are for apoptotic nuclei, or they correspond to apoptotic nuclei. So these are nuclei that are going, or nuclei of cells that are undergoing programmed cell death. And one of the main goals here, why this image was taken, is to look at the ratio of the normal and apoptotic nuclei. Why exactly is, of course, taking us a bit too far now, but you can easily imagine, this could be used to study the effect of different compounds and so on. So the actual goal that we want to analyze in this image is sort of count the number of normal and apoptotic nuclei. And for this, we now need to distinguish them, right? So we basically need to come up with an automatic method to tell them apart from each other. And again, we can just solve this with a machine learning model, basically saying, okay, we take each object, each segmented nucleus, and then classify it into the ones that are normal and the ones that are apoptotic. And Elastic, so the software that we've used before for segmentation, allows us to do the object classification. So what we need to do to distinguish objects into different classes as well. And this works in a pretty similar way as we've seen before. So we again extract features, but these features are now not on a pixel level, but are computed for each object, right? And these encode things like the shape of the object, might also be the location if it's relevant for the classification task, and the intensity distribution, right? And you can already see that shape and intensity would be very helpful for tell the nuclei we want to tell apart. And then these are taken as input to our machine learning model. This is again a random forest as in the example before. And then we have object level annotations from the user to train this model. So basically, as we see here, right, we sort of could interactively now also give annotations for the objects in there, right? Basically telling for all the yellow ones here that these are the normal nuclei, and for the blue ones here that these are the apoptotic nuclei. And then we train the model based on these annotations and with the object features and apply the corresponding model and get predictions for all the nuclei. And now, as you can see here, we can accurately tell all the nuclei apart now. And again, you might have been able to define all of this with rules by just looking at the intensities and the shapes, but it's a lot simpler here to just do this based on training data and let the machine learning model figure out how to separate the nuclei with this. And for more complex and difficult tasks, right, it would be very hard to do this with rules, and it's more efficient and works better to do this in a learned way with machine learning instead. Yeah. So we've seen now two problems in image analysis, one for segmentation, one for object classification, where we can use machine learning. And this is, I hope you see now, a very powerful concept for analyzing images. Questions now, right, that we've seen that we can use kind of normal machine learning for this. Why do we actually need to deep learning, which will be the main topic of the course? Well, we already looked at these more complex problems here, right, sort of the segmentation of mitochondria in EM, or the segmentations of cells in phase contrast. And these are a bit harder problems than what we've seen with the nuclei in phase contrast. I think you can immediately see this looking at the image, right? Sort of separating these touching cells in phase contrast, where we cannot use the intensity as such a clear signal, will be much harder than separating these nuclei in fluorescence. And also here in EM, where we sort of have intensity values for all kinds of different structures, this segmentation task will be harder. And for these kinds of tasks, it's difficult to find a good feature representations. So in other words, by just computing these image filters, as we've done before, stacking them for the pixels and using this for the machine learning algorithm will not really give us very good results. We'll not go through this, but this is unfortunately the case. So for this, we need a more powerful model. And this more powerful model is deep learning. And the idea there is that we don't need to define these features anymore, right? So basically, we are not computing all these image filters as input for our model, but instead the deep learning model is applied directly to the image and can also learn these features together with the decision from the data. And the way this works is by having a much larger model, right? So these deep learning models, they have much more parameters than the machine learning models like decision trees we looked at so far. And so they can learn these features themselves. So this is more powerful, but the downside is that we will also need much more training data to fit them to the data. But if we have this, they will allow us to solve much more complex tasks than the easier machine learning approaches that we've discussed now. Yeah, so this was everything to cover for the basic lectures, right? You've seen the concepts of machine learning, image analysis, and how they were applied to, how machine learning was applied to image data. Again, we'll provide a few more sources in the video description, and then we have covered everything that you should know before the course. In the course itself, we will then delve much deeper into deep learning, how it's different from machine learning, and how it can be used for image analysis application to solve your problems on your image data.